Great to see so many people here today. Well, this is a little bit of a mid-year update. We crammed a lot into our London Calling talk just a couple of months ago, and I had a really hard time actually trying to pare that down to the things that I hear coming up again and again in terms of questions for progress. So hopefully this mid-year report will help you. Everyone that works in Oxford Nanopore R&D, we are all really geared towards this vision that we have of enabling the analysis of anything by anyone anywhere. And that is what our teams get up in the morning and try and do. We provide technology updates every year. We want to bring the whole entire community along with us as we go through this journey. It's been a long journey, but it is at an incredibly exciting point just now. So the big thing that we've really been talking about for the last year is Q20 Plus. Many of you will know this as R10 and Q14. And this is really delivering your biology in unprecedented definition and scale. And the quality of the talks that we've heard today and over the last few days and all the publications that come out are just really inspiring, very exciting. And it's great to see the biology that you guys are achieving. So there's a massive breadth of methods and approaches that people are using, from genotyping to standard whole genome sequencing. What used to be assembly isn't really assembly anymore, and of course the TDT assembly that we've heard a lot about over the last few days. And that is the breadth that you can achieve with this single platform, with this one technology, and it's what we're really excited to carry on developing for you. We're trying to simplify everything at Oxford Nanopore this year. When we launched Q14 to start with, it ran at 2 different speeds, there were 3 different algorithms, and then there was 4 kilohertz and 5 kilohertz. At some point there was about 17 different ways of running one thing, which is too much. So you will start hearing us really break it down into the simplex mode of running and the duplex mode of running. So Q20, Q30, those are the 2 modes of running Nanopore sequencing. So on to simplex. It is a Q20 Plus performance. The release that we did earlier this year in Minnow, we pushed up our rates up to 5 kilohertz, our sensing rates. That let us achieve Q20 Plus even when we're just running high accuracy base calling. That means that everything gets a lot simpler. You don't need to start making that decision. Do I run fast? Do I run hack? You can run hack and you get 99%. SUP is there if you need it. We have established workflows now. Everyone should be getting towards 100 gigabases of data on the Promethon flow cell. If you're not, please reach out to our support teams. You heard this morning, we've got 70 people now in APAC. Please reach out to us. We want to talk to you. We want to understand why, because we've really worked hard to get these established workflows so that you can generate this type of data. Regarding variant calling and variant accuracy, a lot of talks already this morning, and Dan at lunchtime, he clearly showed that with the Q20 Plus data, you can see Snips, you can see indels, particularly in the coding regions, structural variants, best in class, and there was a great paper earlier this year from the NIH card that from a single Promethon flow cell, they were achieving similar, if not better, SNP performance than short-read sequencing. So what we see, the community then replicate, which is wonderful. This technology should also be delivering, for you guys, the best modification quality. So our DNA mod quality in CpG context is over 99%, particularly for 5-methyl-C. We also have 5-HMC, and our algorithms teams will be launching new versions of this so that you get over 98% accuracy with 5MC, 5-HMC. Again, publications show that the benefit of running Nanopore is that you can get the modification, but you can also get haplotype-specific results. And this is always really exciting biology to see. So DNA mods are not limited to CpG context. There's more. And our teams are working constantly on trying to extract every last modified base from that information that we get from our sequencer. So fully integrated in Minnow, so touch of a button is 5MC and 5HMC. Currently, open source offerings in GitHub are all context 5MC, all context 6MA. You can run them post-run via Dorado as well, and we will be integrating these into Minnow by the end of this year. So we will get all context mods running off your box. Consensus. This has always been a really big theme. We've actually been doing really, really well at Bacterial Consensus, really since the early days. And the publication by the Mads Albertsen lab must have been around 2 or 3 years ago now when they first used R10, showed really clearly that you could get a near complete genome. No short read polishing, that's a thing of the past for bacterial assemblies. But for us, near finished is not finished. So our teams have been working actually quite hard. We have been working with our users who, some of them work with bacterial strains that have a lot of interesting modified bases, novel motifs. And of course, we're sequencing native strands. So any new modification is going to throw our base callers. So the team have established really, really great relationships out in the field. We've built a fantastic cohort of samples. We've been looking at them and creating a new bacterial model. That model will be in Rio over the next couple of weeks, so anyone who is doing a lot of bacterial sequencing and wants to have a go, please use it. But you can see what we've managed to do is significantly push further the consensus accuracy of bacterial assemblies, particularly with strains that have interesting modified motifs. So then the next thing to do is obviously put everything in a box. Everyone here wants to get in a lab, switch on the sequencer, load a sample, press go, and they want base called files out the other end. They don't want to bind other computes. They don't want to set up big compute centers. You want it all in the box. And what we've managed to achieve this year is to integrate Minnow with our accelerated base callers with Dorado. And a Promethon A-series tower can now keep up with over 36 flow cells. And you can see that is with Simplex, Simplex plus mods, Simplex mods and alignment. And we will continue to push this. We will get 48 flow cells in real time. So Dorado is really coming into an age of its own. It's getting fully featured. It was really beneficial for us with Promethon, but it's also put Mac back on the grid for Minnow. So we've got the return of the Mac with the latest release of Minnow. So anyone who remembers back in 2014, our Minnows could perfectly well plug into a Mac. And then unfortunately, we started to generate an awful amount of data, so much data that the Macs couldn't keep up anymore. So with the new silicon that Mac have come out with, we can finally keep up with Minnows, and we're really excited about that. So with base calling starting to get under control, we can now really start accelerating the scalability of Promethon. And remember, P2 Solo is available from only $10,500. And for that, you can be sequencing 1 genome per flow cell at $690, pushing towards 2 genomes at $345. And we will go further. We've done it before. We've managed to increase massively our outputs, and we will do it again. And we will get genomes at $235 and sub $200. And everything on the same device that you bought today. We get a lot of questions about storage. We get a lot of questions about file types. Oxford Nanopore is the only company at this point in time that lets you keep your raw data. And we do that because we've been on a journey with everyone in the community, and as we improve our algorithms, people like to recall their old data. As we uncover new modification signals, people like to discover new things. So, but if you look, we also now produce, with the base calling in real time, very, very standard file formats. So the fast queues, BAMs, VCFs, they are all perfectly perfectly normal size files. During 2024, we will, of course, make everyone aware, and we will be writing to everyone, but we will switch the default mode to being raw read off. But you will be able to select, if you want to keep your raw reads, you will be able to select that at runtime. We've had a lot of questions about storage. How much does it cost to store all of these different file formats that we're generating? And the teams have worked with AWS to get a rough estimate. And if you're storing things in S3, it's about $12 a month for raw data, or $1 a month if you go glacial. So there's lots of options there. Once you've generated the data, of course, you need to analyze it, and you want to analyze it. And I know the workshop yesterday for Epitome was absolutely rammed. And we are working tirelessly on the Epitome platform, making it ever easier to use and more complete with more workflows. So moving over to Duplex. There was a question earlier today, I think it was during JJ's talk, of how do you get Duplex. So Duplex is when you sequence the template and complement strand together, and you sort of pair them back up for higher accuracy. It produces Q20 Plus accuracy. That's what you get when you sequence Duplex. We showed at London Calling, we showed that on native human samples, these are all internal, highly clean and highly pure samples, we were getting over 50 gigs of Duplex from a single Promethon flow cell. And remember that that also has an additional 40 gigs of simplex data alongside it, and simplex data is Q20. So you actually get 90 gigs overall of data, which is above Q20 with a big peak of it at Q30. We have a lot of headroom in the system. We can see that with purified and amplified E. coli, we can get over 100 gigs of Duplex, and that is what our teams are working on, is how do we bridge that gap, and how do we get all of those solutions into the hands of our users. So Duplex has become really exciting, really because of the dawn of the telomere-to-telomere genome. We've been discussing in a lot of the meetings today, assembly used to be a sort of a once-in-a-career occurrence. It's a little bit mental that you've now got projects that are attempting 300 T2T genomes, 1,000 T2T genomes. So this is, it's just really exciting to see what can come of a massive collaborative effort. This is Karen Miga, this is her HBT talk from February this year, and she was really explaining that the initial T2T came together, it was multi-center, multi-platform, but if we're going to accelerate the generation of telomere to telomere, it needs to become simpler, it needs to become much easier to use. And so therefore, there's the drive to try and get it all onto one platform. And JJ did the talk all this morning about his results. But what we're really excited about, especially with the dawn of the P2 era, is that you can now do this anywhere. And the T2T consortium are really excited to be broadening access to this type of analysis, this type of assembly, and we really want to do this anywhere. Internally, our applications team have been working hard to figure out what are the best recipes. Internally, we're running 40x Duplex, 40x Ultra-Long, 40x POC. I think it's quite similar to the recommendations that we heard this morning. And how do you get it? So high Duplex, it is a different flow cell, it's a different run condition, and so you can register for early access. And we have a registration-based early access, so you need to register on the website. Once you register on the website, we let you into an early access shop where you can get the product. And we've got it in the hands of quite a few users today. We're working incredibly hard to get high Duplex out in the open shop. We think a really big piece of that will be the integration of Duplex into Minnow. We think that's going to be really key in order for customers to be able to run it easily. And so we will be doing that before the end of this year. So the overall chemistry timeline, we are in the middle of a chemistry transition. So of course we've done Promethon, we did in Q1 this year. We've done Minnow, Gridiron, Flongle. All of those R1041 flow cells are done, they're released. We will be looking at putting the high Duplex flow cell during Q4 into broad early access. All the Kit 14 types are in the release bucket. We've got the cDNA and the 16S kit to do at the end of this year. And we've been working with the community to transition away from R9. And on Promethon, that's been amazing. Over 90% of runs now are on the latest chemistry. A lot of our Minnow users need a little bit of extra help, so we will be extending the availability of R9 until the end of Q1 next year for our Minnow users to give you a little bit more time to transition. Please do keep an eye on the community. We are posting there very, very regularly, be it updates about discontinuation timelines. We're also constantly getting feedback from the field about how the R10 implementation is going, and we're putting up best practices, tips, how to get the most out of your runs. So please keep an eye on the community. So the next hot topic that we get, and a lot of excitement that I see is RNA. And if it's not direct, it's a copy. And if it's a copy, that's fine. We can sequence it as well. But really, direct RNA, we are the only platform who can do it. We have an RNA direct reader head. We've got an RNA-specific nanopore, an RNA-specific motor. The wonderful thing that you get is no transcription bias, but also you get modifications. It's a really amazing benefit of the direct RNA system. So we are moving from this RNA2 to the RNA4. So new kit, new flow cell part numbers. And what users are getting out of it is, with that increased speed, we're seeing 4 to 5x improvement in output. We've been really working a lot on RNA now on the Promethon flow cell as well. When we first launched it, it was really sort of a Minnow-driven application. But with the latest outputs, Promethon is coming in really well. So this is giving us about 30 million reads on a Promethon flow cell. This is, again, it's a very clean control sample. And remember, if you need more than that, if you're really doing expression analysis, we have the cDNA range where you're getting 100 million reads plus. But for direct RNA, 30 million reads, you can actually start looking at getting, with that increased transcriptome coverage, you can start looking at better representation of your sample. And you can see here that you've got really great concordance through the increased read depth. The really exciting thing, and please do talk to Adrian, he's got a poster in the room next door, is that we're going to be releasing our first RNA modification model. So this is a native M6A model, and it will be able to deliver sort of unmodified, a true negative rate of 92% accuracy, which is miles ahead of any other technology that you have available right now to look at RNA modifications. Our algorithms team and our RNA mods team, they really, really want to work with the community. So please do engage with us. If you have interesting projects, if you have interesting samples, if you have truth sets, please do engage with our team, because we really, really want to hear from you. And again, on the RNA side, we have a closed early access. It's active now. So again, you can register on our website, and you can gain access to the RNA Store. We've talked a lot about T to T today. I'm going to talk a little bit about E to E. These are the end-to-end workflows that we are pushing together. We hear a lot of feedback from the user community. If you go online, there are a lot of protocols, there's a lot of advice. So we have probably about 150 different protocols online. We've got protocol selectors, protocol builders, getting started guides. If you then go into the extraction, we have over 72 sample protocols so that to help you get the most out of your sample type. And again, if you look at Epitome, it's a very, very rapidly growing list of bioinformatics analyses that you can now do online. So what we've done is effectively we've open sourced our platform. We have given you guides on how to get the most out of it, and everyone is finding their own path, finding their own way to get through the applications. And what we've started to do really is to go from this open source to lockdown recommendations. We had a lot of customers coming to us to say, I want to know exactly how to run your machine so that I get the same result each and every time. I want to stop exploring. I actually want to get some results out the same every day of the week. So for this, we've started to increase the number of these end-to-end protocols that we have online. Things like the human workflows, they have minimum output guides, and then we have reduced representation methylation sequencing, plasmid sequencing, 10x, all these different applications that you can now go online, and they have sample, extraction, library preparation, sequencing, analysis, all in one place with some really, really helpful walkthrough guides. So we do innovate. We try and innovate with care. So in our life science research tool products, we have developer, early access, and release phases. We have Q-Line for anyone who's trying to develop their own assays and needs a lockdown product. And we are also working on applied market and partner products. But we do continue to innovate. And those products will go through, again, this sort of careful release stage gate from developer through early access all the way through to released, and the right products ending up in our Q-Line. And so with that, I think I've managed to do 40 slides in 20 minutes.